So digestion starts by just stick it in your mouth, basically, insert it into the oral cavity. Then all these different processes start to happen. For example, what's meant by mechanical digestion that refers to just the physical pummeling of food. And that's mostly accomplished by, the choppers in your mouth. Your teeth, call them choppers. That's what first happens in your mouth. But also the stomach churning accomplishes a lot of that. The stomach's like a blender. I would say that's the basic idea. Just pummel the food. The result of chewing your food in your oral cavity, the result is, they call it a bolus of food. What food looks like right before you swallow. When it's ready to swallow, it's been chopped up and solubilized with saliva. In the stomach churning, you add acid to it. You create this acidic chyme. Just by pummeling the food. But then, to really break up the food to absorb at the molecular level, you're going to have chemical digestion. Before you can have that, you really need to find, just to go over propulsion. I'm going to say peristalsis. That process involves rhythmic contraction in the smooth muscle layer of the GI tract wall. Rhythmic contraction of the smooth muscle layer. In GI tract wall. In case you don't know, GI is gastrointestinal tract. That's basically what we're talking about here. So gastrointestinal is GI. The tract is a tube from mouth to anus, and that's one of the things you see here. And peristalsis is illustrated by this greenish line, and it starts from the beginning and it goes all the way through to the end. So from beginning to end, the goal of peristalsis is to push the food stuff in one direction towards the anus. Pushes food in one direction towards anus. And we'll take a look at those smooth muscle layers. There's actually multiple layers in another slide, but just the idea of peristalsis, because once you swallow your food, this takes care of itself. So chemical digestion is true digestion. Meaning that true digestion in the sense that you're breaking down the molecule, you're breaking down the food at a molecular level. Polymers into monomers. So you have to think about what that means when you eat starch, complex carbs, and simple carbs. You know, like sucrose and all the sugars that you eat, the pastas and everything else. Proteins in meat or plant products, you break down into amino acids. The fats, you break down the triglycerides into like glycerol and free fatty acids. Well, this chemical digestion actually begins in the oral cavity because saliva contains salivary amylase, which can start to break down some of the starches. So, oral cavity has salivary amylase. Then you swallow the food and it gets down to the stomach. And the stomach is another key organ for chemical digestion, mostly because of the stomach acid. Good old HCl is a very effective chemical digester. There's other things in the stomach, for example, pepsin from the stomach. The peptide bonds in proteins are broken by this molecule. That comes from the stomach. So, and then, once food gets past the stomach, you have like about 20 feet of small intestine to finish off chemical digestion. There's something called brush border enzymes in small intestines. Small intestines have what are called brush border enzymes. Now the brush border enzymes, they kind of line the microvilli of the epithelial cells, and that's why they're called brush border enzymes. The microvilli appear like a brush border on the microscope. So that's basically most of the chemical digestion. I should also mention that there are things that are secreted into the digestive tract by mostly liver and pancreas. So if you look at the figure, they symbolize chemical digestion by these acid drops. And most of the acid drops occur right around here in the first part of the small intestine called the duodenum. The duodenum receives secretions from, was not shown here, but it receives chemicals from the liver and the pancreas. Okay. So there's something called accessory organs of digestion. Accessory organs of digestion, they're not a part of the GI tract, but they secrete things into it. And that's liver and pancreas, those are the main ones. I mean, technically your salivary glands are accessory organs because they're not in the tract, but they secrete things into your oral cavity. So the accessory organs are liver and pancreas. The accessory organ, what they secrete is the pancreas. They'll secrete a combination of bicarbonate and enzymes. Bicarbonate is, HCO3 negative charge enzymes. They'll secrete things into the digestive tract at the duodenum. The duodenum is about the first 10 inches of the small intestine. The small intestine is again about 20 feet, so just the first 10 inches. Because think about it. if you're the first part after the stomach, you're receiving all that acidic chyme. So the bicarbonate will help neutralize it, and the enzymes will help continue the chemical digestion. The liver will secrete into the duodenum bile. Bile will help emulsify the fats for fat digestion. So both are very important for chemical digestion. And then, once the food molecules are small enough to be absorbed, that's accomplished mostly by the small intestine. You can absorb things along the way in other parts of the digestive tract, like for example through the stomach mucosa, but mostly absorption of things is by the small intestine, simply because it's so long. It's a tube that's about maybe 1 inch in diameter and about 20 feet long. Okay. And the small intestine is well equipped for absorption, and you absorb the food molecules into intestinal capillaries, but also lymphatics. And the name of the lymphatic vessel in the capillaries is called the lacteal. So lacteal are lymphatics. Lacteals are found in the small intestines in these little finger-like projections called villi of the mucosa. I'll show you a picture of that later. You need the lacteals because the fat globules of the fat you eat, those are too big to get into the intestinal capillaries, the blood capillaries, but lacteals can absorb them. So that's why they show an intestinal capillary blood vessel and a lymph vessel. That's where most of the absorption takes place. So you absorb along the small intestine as well as the large intestine. However, mostly it occurs during the small. So the small intestine is followed by the large intestine. The large intestine mostly absorbs maybe water and fat-soluble vitamins. Whatever's left after the small intestine is finished absorbing most of the food. Water, fat-soluble vitamins. And basically, by absorbing water, you compact the feces. Essentially, what feces are all the things that could not be absorbed by the digestive tract, things like fiber. Okay. And by the time you get to the end of the digestive tract, you're at rectum anus. So defecation, it's a reflex involving the rectum anus. Think of the rectum as the end of the digestive tract. is basically a holding chamber for feces. The rectum is a holding chamber for feces. What happens is the feces will distend the rectal wall. The reflex is it to contract in response to being stretched. So stretch, contract, but then you push the feces toward the anal canal. And that gives you the sensation that you have to go. So rectal wall distends, that means stretches out, contracts. Then as the food approaches the anal canal, you have to make the decision if you want to, go or not, because there's sphincters there that keep it shut. There's the external anal sphincter, that skeletal muscle that you control with conscious thought, right? So you can voluntarily hold it or decide to go. So I'll write that one. External anal sphincter. Is under voluntary control. Technically, you could take like a 30-foot string, swallow it, and you should be able to pull it out the other side. This tube is continuous, okay? And so we're going to talk about it from start to finish, but it helps to kind of first understand the general structure of the GI tract. When we talk about the histology, we'll look at different parts of the GI tract. For the histology, you're going to have to be able to look at the GI tract wall of the esophagus, the stomach, the small intestine, 3 parts of the small intestine, duodenum, jejunum, ileum, and then large intestine. That's 6 different locations of the GI tract that I should be able to show you, and you should say, oh yeah, that's the stomach. Oh yeah, that's the duodenum. Oh yeah, that's the ileum. So I have to teach all that. But it helps to understand the general structure of all of those locations, because they all follow a general pattern. So that's what we have here. General pattern of the GI tract wall. So think about this illustration when we study histology. I'll probably present the histology of the GI tract next class. The general pattern of the GI tract wall can be understood by studying the different layers. Mucosa, submucosa, muscularis externa, serosa. 4 basic layers. We'll go from inner to outer. The lumen is where food is passing through it, but we're going to study the wall that surrounds the food passing through this tube. Okay. So mucosa is the innermost layer. This is the layer that's kind of moving. That's mucosa, not mucose. It's the layer that is agitating the food. It's touching the food. Innermost layer agitates passing food. No matter where you walk. Okay, innermost layer. And it consists of an epithelium, which kind you have to know, but I can't really tell you now. It depends where you are in the GI tract. The function that's required will determine the epithelium. Generally speaking, at the beginning and the end, right? The oral cavity and towards the anus, it's a stratified epithelium for protection against abrasion, because things is rubbing by it as it food enters and food exits. Everywhere else is pretty much some kind of columnar epithelium for the function of secretion and absorption. So basically, for now, I'll just say the epithelium changes depending on function. Deep to that, going from inner to outer on this figure, the epithelium which changes is just the connective tissue that underlies the epithelium is called the lamina propria. And that's just the loose connective tissue. And surrounding the lamina propria, what's unique about the GI tract is the mucosa has its own muscle layer. It's called the muscularis mucosa. And, well, having the muscle layer close inside increases the agitation of the food, which is good for digestion. By doing that, you're going to move food along better too. Okay, so that's it. That's pretty much it. I would put a star next to the muscularis mucosa. You usually don't see that when you study histology. But in the GI tract, epithelium, lamina propria, muscularis mucosa, those three sublayers within this layer is what you should be able to identify. Surrounding the mucosa is the submucosa. More loose CT. There's nothing much to look at. There are nerves in there that make a nerve plexus. We just call it the submucosal plexus. Anytime you see yellow illustrated within this connective tissue layer, right here, you're going for submucosal plexus. The submucosal plexus, well, it's part of the enteric nervous system. I have a slide on that later, okay? But also, in the duodenum, I want you to be able to identify these glands called the Brunner's glands. They do illustrate glands here in the submucosa on this picture that will secrete into the digestive tract. The ones I want you to know are called Brunner's glands, pronounced Brunner's, spelled Brunner's. The Brunner's glands will secrete an alkaline fluid in the duodenum to help neutralize acid in the duodenum. So they're in the duodenum, that part of the digestive tract. They're in the submucosa. Secrete an alkaline fluid that'll help neutralize the stomach acid. Surrounding that is the main muscle layer of the digestive tract called muscularis externa. It's external to the muscularis mucosa inside there. And it's double layered most of the time. So muscularis mucosa, not to be confused with muscularis externa. usually bilayered. And the orientation of the smooth muscle cells in the two layers are at perpendicular angles. One, the smooth muscle layer are arranged circularly around the digestive tract. See this layer here, this inner circular layer? They're arranged in this plane. So you have an inner circular layer. You don't have to put inner, but it's called circular layer. The circular layer by itself means nothing. You always have to put of muscularis externa. And by being a circular layer, you can kind of pinch shut the digestive tract. So you can kind of, if you're a tube, you can kind of squeeze like that, help push the food forward. Okay. Now the layer surrounding that is a longitudinal layer. It's along the length of the tube. That's the orientation of the smooth muscle cells here. So you have the outer longitudinal layer of muscularis externa. So that'll allow you to scrunch the tube, kind of like scrunch it that way. Right, so you can kind of pinch and scrunch, and that'll help push food in one direction. That will accomplish peristalsis and other things, but the bilayered, so usually bi's bad grammar. I'll say it's usually bilayered, and that it accomplishes peristalsis. So I say usually bilayered. The exception is the stomach. The stomach has, it's tri-layered. There's 3 layers. Stomach only. There's a 3rd layer. It's called the oblique layer. It's kind of like you have a 3rd layer that kind of runs obliquely to these other 2 layers. So you add the oblique layer in the stomach, and it's 3 layers total. Everywhere else, it's 2 layers that you should be able to identify. Stomach, be able to identify 3. Now to wrap it all up, you have the serosa or adventitia. Serosa or adventitia, terms are interchangeable. It's the outermost layer. It's basically it's an epithelium and connective tissue. It's a mucus. It's a serous membrane. Serosa. It's a serous membrane. Serous membrane. The serosa form mesentery. The mesentery are kind of the boundaries and structures that hold all the digestive viscera in place. Okay. What's inaccurate about our models is that they give you the impression that like your insides are like a Mr. Potato Head. You can just take off all the things, all the parts. Oh, look at this. Just put them all back in. Your organs are all connected. And not only are they connected to each other, the mesenteries hold everything in place by this serosa. So for example, so you have like the back body wall, okay? and you have part of your digestive tract that's interior to your back body wall. The serosa, it also lines your back body wall. It's called the peritoneum. Sometimes the serosa will kind of like shoot out and wrap around the intestinal organs. So my blue line represents this serous membrane that formed the mesentery. The mesentery is where you have this double pinching right here. The part that wraps around the organ is like a visceral peritoneum. That's kind of what it's called. It's a serosa, but we can also refer to it as the visceral peritoneum. This serosa that lines the body cavity, say for example the back body wall, is called the parietal peritoneum. So the serosa that I just defined is essentially also the visceral peritoneum, one and the same. It's that outermost layer. And you can see that on the figure, it wraps all the way around, and where it pinches on this side here, it's the mesentery. And you can also see that the mesentery aren't empty. The double membrane contains nerve, artery, vein, and lymphatics. And that's kind of the delivery system for those systems along every inch of intestinal tract. Okay. That's really it. Those basic layers, like for example, we have here, oh, this slide reminds me I forgot one thing. The thing I forgot is in this layer here. Add to muscularis externa the myenteric plexus, another plexus of nerves along with submucosal plexus. You usually see nerves of the plexus stuck between the layers. Here it is here. Okay. On the previous slide, let me go back and show it. Here it is here, myenteric plexus between these two layers. Well, anyways, what I want you to know is that the myenteric plexus, in addition to the submucosal plexus, comprise the enteric nervous system. Think of it as mini brain. As soon as you swallow food, the only thing you realize is when you feel the urge to go at the other end. Everything else in between is unconscious. And it's all taken care of by this nervous system. Think of it as a third nervous system. We do a good job teaching the autonomic and somatic, but we really don't teach this one very much. It's basically the nervous system of the GI tract. So myenteric plexus plus submucosal plexus equals E and S, the enteric nervous system. Here's a picture of a model that we have in the room. I want you to study for the lab practical. I just have bracketed the different layers: mucosa, submucosa, muscularis externa, and the serosa on the very bottom. It happens to be small intestine. I mean, it could have been any other part of the digestive tract. I'll talk about this a little bit. Number one, those are all mucosa, but the mucosa is thrown into this shape. These finger-like projections are called villi. And if you look on your notes, I said the intestine accomplishes most of absorption through intestinal capillaries and lacteal, and those can be seen in a number of places. Number 3 points to intestinal capillaries. Number 2 points to lacteal. I can also have you identify this over here, even though I'm not pointing to it. Those are also intestinal capillaries. Okay. Let's see here. Number 4 is pointing to the gray. Okay, so you have this epithelium, which is a columnar epithelium, but all the gray is the lamina propria. Let's see, I'll just do all the numbers for now. Number 15, that's an intestinal crypt. Maybe I should teach some of this stuff when I get to the small intestine. Let me present the things that you know. You should know number 9. It's the outermost layer of the mucosa. It's the muscularis mucosa. So from here to here is submucosa. And number 10 is the Brunner's glands. So you can assume this is duodenum. Number 11 I taught you, MALT, mucosa-associated lymphoid tissue, this big lymphatic tissue structure. So it's basically in the submucosa and invades the mucosa. It's mucosa-associated. So this is all submucosa. 7 is pointing to the yellow, that's the submucosal plexus. So 13 is the myenteric plexus, and you should be able to differentiate between the 12A and the B. It's the inner circular and the outer longitudinal. And then right here is the serosa or adventitia. Now these different layers are shown in different models, and here's a more simplified picture. It's a little blurry, but just follow the order. So mucosa, submucosa, and then both layers of muscularis externa, the circular longitudinal. And then that color is adventitia. That's a pretty accurate color of what intestines look like in a cadaver. So for the large intestine, you can see the whole model here. I just kind of zoomed in to take this picture here. Mucosa, submucosa, the circular layer. Notice in the large intestine, the longitudinal layer is not a full layer. It's reduced to like these 3 little ribbons. Here's one. You have these ribbons all like in the front and the back, and the ribbons are called tenia coli, but you can think of it as the longitudinal layer of muscularis externa, just reduced to a ribbon. And then so 11 would be the serosa or adventitia. So I want you to know how all the digestive organs are held in place by the mesentery. I kind of already described this. If you have the back body wall lined with a parietal peritoneum, the part that kind of like shoots out to wrap around the organs, that's the mesentery. But the layer that is intimate with the organ is the visceral peritoneum. Now some organs in development are pushed behind the body wall, and they're referred to as retroperitoneal, like this structure back here. If it's in the body cavity, like this or this, you say intraperitoneal. So there's intra, and then there's retro. Okay, so I'll be using that term from time to time. Intra versus retro peritoneal. Well, here's a cross-sectional view. It's a sagittal section, not a cross-section, I should say. This was cross-sectional view. There's the vertebra and back. This is a sagittal section. And it'll kind of like take us through all the mesenteries I want you to know. Most things here are intra. I'll show you what's retro. So I can kind of like get a little sketch. I'll make it a little bigger so I can draw. But if this is the kind of like the sketch of that, what I would do is I would draw some of the intraperitoneal organs like liver. The liver is underneath the muscular diaphragm. The diaphragm is a physical membrane that separates the thoracic and abdominal cavities where most of the digestive tract organs are. Underneath the liver, you got stomach. And underneath that, you got the transverse colon. And then you have a bunch of cut small intestines. Notably, the jejunum and the ileum is most of the small intestine. The first part, the duodenum, is up here. They're all connected, but in a sectional view, it looks like they're disconnected. But the duodenum and pancreas are retroperitoneal, behind the stomach and transverse colon. Pancreas, and then part of the duodenum. And then a bunch of small intestines. So all these things are loosely attached to each other in mesentery. If you kind of start this by drawing over the pancreas, which is retroperitoneal. So my blue line is like the parietal peritoneum. And it lines the back of the body wall. It lines the inside here of the diaphragm. There's a part that shoots down that goes over the liver on one side and the other. So notice that there's a little bald spot of the liver where it's not covered with the mesentery, with the visceral peritoneum. And they literally call that the bare spot of the liver. Okay, anyways, this mesentery wraps all the way around this peritoneum wraps all the way around liver. And there's a part of mesentery that will shoot down and then go over the stomach. And so that little bit of mesentery is called the lesser omentum, the one between the liver and the stomach. Then there's this big fat apron that comes off of the stomach. Anyways, it forms like a little apron, goes back up and attaches to the transverse colon. So on my drawing on here, I'm just following this big fat apron right there, wraps up to the transverse colon. It's called the greater omentum, the fat apron. All of this, what I drew here. greater momentum. Well, anyways, the transverse colon is anchored to the back body wall. There's mesentery that will shoot back. Go with the back body wall. Anyways, this is called, that mesentery is the transverse mesocolon. Transverse mesocolon. Try to draw it in there. And then you have mesentery that wrap all around the small intestines. This mesentery is called the root of the mesentery. I can just kind of finish off the rest of the parietal peritube. That's basically, that's really a more accurate picture of how the organs are fixed in place by mesenteries. There's other mesenteries. I think I gave you all the main ones I want you to know. And I have some more pictures to show you the mesenteries. Like, for example, here. Lesser omentum, greater omentum. They're named after their attachments to the stomach. Now, the stomach has these curvatures called the lesser curvature attached to lesser omentum, the omentum between liver and stomach, and then the greater curvature has attached to it the greater omentum, which like forms an apron over the intestines there. So if you lift up the fat apron, as I call it, the greater omentum, you can see how it attaches to the transverse mesocolon underneath it. And then by doing that, you can see how the transverse mesocolon proceeds to attach to the back body wall. There, and then here. Okay. So here's a model we have in the room. There's the duodenum, which is along with the pancreas, both are which partially retroperitoneal. There's the beginning of the duodenum. There's the end of it. Okay? And there's the transverse mesocolon. Now, the root of the mesentery can be seen on the picture on the right. Think of the small intestines here being cut. They cut the tube there. It would be along the whole length of this mesentery. It's like you gather it up and attach it to the back. That's the root of the mesentery, right there. All right, so that's kind of the mesenteries. And if you kind of take a step back and look at the whole GI tract, let's go back and start with the oral cavity, or mouth. You can breathe through your mouth, but again, the oral cavity is not part of the airway, it's the foodway. So this is kind of where we start. And they kind of start with a Dennis view of the oral cavity shown here. I don't like where they put oral cavity proper. The oral cavity in your mouth is the space between your tongue and your palate. An oral cavity proper, simply your mouth, the space between tongue and palate. The roof of your mouth is your palate. The only other thing I wanted you to know was what's called the oral vestibule. They point to your gums, but it's the space between basically your gum and lips or your gum and cheeks. Oral vestibule. So the word for gums in anatomy is gingiva. And the word for lips in anatomy is the labrum or labia, since you have two lips, upper and lower. However, if it's the cheeks, the word for cheeks is buccal. So I'll refer to it as a region, the buccal region. Buccal, cheeks, yeah. Labrum, lips. That's basically where you put a mouthpiece, a U-shaped mouthpiece, in the oral vestibule to protect your teeth. So in terms of oral cavity, I'll teach the structure of teeth as well as the salivary glands. Oops, it's not cooperating. The salivary glands, there's 3 of them, and they're all shown here and there. And I want you to know the cranial nerve innervation. You have the parotid gland, and since these are all exocrine function, they have ducts too. But the one duct I usually have you identify on lab practicals is the parotid duct, because it's the longest. You can see it here. Okay. The superficial is on top of some of the muscles, and the duct has to pierce the buccinator to get in the oral cavity. And they're kind of empty like by around the 2nd molar of your upper jaw. So, salivary glands. Parotid salivary gland. And you have submandibular salivary gland. And then sublingual salivary gland. The innervation is 977. That's the glossopharyngeal and the lingual nerves. So I would also note, be able to identify the parotid duct as well. Now the other ones have ducts too. Usually you can't see them on models. You can look for them, but all glands have a duct. Otherwise they're useless. You have to empty into somewhere. The parotid duct you can usually see. That's the one I said. I should probably write what I said. Pierces buccinator. One of your mouth muscles. That's the muscle called the musician's muscle. It helps if you play a woodwind or a horn instrument, your obture, the shape of your mouth, the blow air through your instrument in a skillful fashion. That muscle is used quite a bit. So I'm writing pierces buccinator to empty in oral cavity. By 2nd molar, upper jaw, Well, what I really want to focus on is saliva itself and the histology of the salivary glands. They kind of differ. Saliva itself is a digestive fluid. It contains the salivary amylase as well as serous fluid with the amylase and mucousy fluid to help solubilize the food. So basically watery mucousy, okay, with a little bit of amylase. I already noted that. And the histology is, if it is a serous saliva, the cells that secrete it in the gland will stain dark. If it's a mucousy type of saliva, the cells that secrete mucus saliva stain light here and here. Basically, hardly have any here. It depends on the duct length. A watery saliva has no problem traveling down a long duct, but mucousy saliva need a shorter duct because it's harder for them to travel through the duct because of the mucousy. And so medium to short ducts can have mucus saliva, but long ducts only have serous. So one way to tell what gland you're looking at in terms of histology is, if it's the parotid, it'll stain dark. Because the cells secrete serous saliva. In fact, it's totally serous. And as I say on the slide, it's because that parotid duct, it's the longest duct length. If I show you a slide where it's like partly dark, partly light, as it is in the submandibular gland, it's because the secretion is a mixture of serous and mucus saliva. So the duct length is kind of, I just say medium length. Shortest ducts are right under your tongue. If you look in the mirror and lift your tongue up and open your mouth, you can see this saliva kind of ooze out under your tongue. That's the sublingual salivary gland. So they're basically light staining because it's mostly mucus. Okay. Sometimes I spell mucus with an O or without, it doesn't matter. I've seen it spelled both ways. So it stains light, it's mostly mucus, hence it's going to have the shortest duct lengths. Shortest ducts. So saliva helps you solubilize the food in the oral cavity, and I should have you know a couple of muscles of mastication. Mastication is the word for chew in anatomy. Temporalis massivum. Two muscles of mastication help you chew. They close the jaw. There's a few muscles that open the jaw. I gave you two that close it. Okay. I'm not going to test you on their attachments. You should be able to identify them at least. And you should still remember their nervous innervation. It's the mandibular division of cranial nerve 5, so basically V3. V3, the name of V3 is the mandibular division. There's a nice picture of it. There's V3 right there. talk about your teeth, your chompers. The structure of teeth, well, if you just look at both jaws, you can see all the teeth are in there. Upper jaw, lower jaw, okay? So in anatomy, we say maxillary teeth, mandibular teeth, right? And the innervation is V2, V3 for sensory function. So if you got teeth, they say dentition is the fancy word for you got teeth. (32 teeth total in an adult. So it goes 16, 16, maxillary teeth. In your upper jaw, the innervation is V2, that's the innervation. Mandibular teeth, V3. Again, so this time it's a sensory function. Like you got a toothache, you feel that, right? So that's part of the nerve of V3. We'll get that in. To name the teeth, think of it as 4 sets of 8, right? A set here, a set there, and 2 sets in your lower jaw. You have 2 incisors. In your set of 8, 4 sets of 8 is what I said. So here's 1 set of 8, 2 incisors. They're not for chewing, they're more for tearing and cutting, right? You have 1 fang, 1 canine. It's a cone-shaped tooth. You have 2 premolars, 3 molars. Sometimes people call your molars bicuspids. The cusp is the chewing surface of a tooth. So 2 premolars, 3 molars. Molars are sometimes called your tricuspids. So that's your set of 8. And here's a picture of, this slide shows V2 and V3. Remember, this is the trigeminal nerve, cranial nerve 5. There's ophthalmic, maxillary, mandibular divisions. But notice the V3, not only does it have branches that supply motor innervation to your muscles of mastication, there's a part of that nerve that enters the mandibular foramen, and it runs through this mandibular canal. This nerve is called the inferior alveolar nerve. And that's the nerve to write down, because branches of it will give nervous innervation to your teeth, okay, in the lower jaw. So for mandibular teeth, inferior alveolar nerve. For upper teeth, there's no one big nerve that I can just name. There's a bunch of little branches that you don't have to know the names of, just know V2. There's a bunch of branches that will supply your upper teeth. Now, in terms of if you need dental work and you need a nerve block, I would prefer it's the lower jaw, because you can easily block that one nerve, so you need less shots, right? If you need work in your upper jaw, they may have to give you multiple shots, because there's not one nerve you can block. You got to block all the little branches. I hate those Novocaine shots. But anyways, you got to block them if they're going to do dental work. Anyway, so that's V2, V3. If you look at a tooth and a mouth, you have different surfaces, these 4 sides of the tooth. So they kind of like, just kind of have this, 4 sides of the tooth. Pick out one of your molars there. Well, the contact surfaces are called mesial and distal. Okay. The mesial is kind of closer to this midline. The distal is just further away from it. It's the side opposite, mesial. I'll just describe it as further away. So the other sides either face the palate or face the lip and gums. So it's either going to be buccal or labial, depending on where you are. But mesial distal, that's the contact surfaces, that's where you floss, in between your teeth. In terms of the dentition, the eruption, and then the shedding, we start with the baby teeth, called the deciduous teeth, because they fall out. Remember, deciduous means that which falls off. So here's where they erupt in months. Okay, incisors come out first, molars come out last, second year of life. And then they're shed, teeth fall out, beginning in, kindergarten, first grade, all the way through 6th grade. And then the eruption of the adult teeth that we're talking about, we're not really teaching the baby teeth here. The eruption in years, you get your first teeth, 1st, 2nd grade, 3rd, 4th grade. Your last molars come out teens, early 20s. That 3rd molar, we call your wisdom teeth. If your jaw is big enough to accommodate the 3rd molar, you got a 3rd molar. Most people like me, my jaw is too small, so before the 3rd molar can erupt and impact the 2nd molar, you remove it surgically. Right? Most of this, raise your hand if you had your wisdom teeth removed. That's a lot of people, right? So it's most people. I had mine removed when I was 14 because it's like, this is going to cause me a lot of pain if they erupt and hit my 2nd molar. So I don't have 32 teeth, I have 28. So I had all 4 of my wisdom teeth pulled out. If you have all 32, go ahead and count it. 1 to 16, upper jaw is self-coated. I don't have 1 or 16, but then drop down to 17, I don't got that either. Go all the way to 32. So I'm missing 1 or 32, 16 and 17, right? And when you extract a tooth, you don't renumber. You skip the number, right? Yeah?
Is it possible to just have upper side the wisdom teeth and not having a down part?
You're saying, is it possible to have some extracted and not others?
Yes.
It just all depends if they're going to cause problems when they erupt. Are they going to bump into the tooth in front of it? If the answer is yes, they'll probably want to have it removed. Okay, that's the periodontist's call. We'll look at an x-ray and find out. Okay, if you cut a tooth open and look at the structure of the tooth, we have this figure. Now, parts of the teeth are really the most mineralized tissue in the human body. They're very hard tissues. And the general structure is crown, neck, root. Now, the crown of your tooth is the part that's erupted above the gingiva. It's above the gum line. The neck of the tooth is where it's kind of the tooth gingiva border. It's kind of where they interact. It's where you floss. tooth gum line border. Then the part you don't see is part of the tooth that's rooted into the alveolar bone of the jawbone is the root of the tooth. So the root is anchored in tooth socket by periodontal ligaments. Okay, they're illustrated there. They bolt the tooth in the socket of alveolar bone. Tooth bolted into socket of alveolar bone by periodontal ligaments. That joint, those ligaments bolting it in, it's called the gomphosis joint, which means bolted condition. Gomphosis joint. You can look up that joint type in that chapter of your book if you want to review that, but that's what it is. And so crown, neck, root, then the layers there. Well, the most mineralized bone in the body is the enamel of your tooth, which lines the crown. Now the enamel is like 96% mineralized. The most mineralized, hardest tissue in the body. Now, if you can see, most of the tooth in crown, neck, root is called dentin. And the dentin is also mineralized, not as much as enamel, it's about 70% mineralized dentin. Now, only in the root, there's this layer called cementum, right there in the root. Cementum is about 50% mineralized. It's similar to bone. So in the center of the tooth, you have what's called the pulp chamber, which is continuous with this canal leading out. Now, the pulp chamber is not mineralized tissue, it's soft tissue. It contains the blood supply and the nerves. Soft tissue. It's got blood vessels. Sensory nerves. So if you get a cavity called dental caries, right? It's an erosion of the enamel through the dentin. If it happens to go far enough to reach the pulp chamber, it can get infected. And when it gets infected, you're gonna have a massive toothache. And if left untreated, you may need that extracted. There's the abscess of the infection. And it's a decision you have to make with your dentist. If you want to save the tooth, you can do what's called a root canal. If you've heard of that. Maybe some of you have had a root canal. Anybody? Was it 2 appointments for you? It usually is. First appointment, they drill and they excavate the pulp chamber. Now what they do is they remove that but they leave the blood vessel, because you don't need the nerve to keep the tooth alive, but you need the blood supply. So they just kind of excavate, first appointment, second appointment, they crown it, right? And yeah, you can still have the tooth, or if he's like, no, we're saving, just extract that. Bad boy, get it out of there. Okay, so that's the tooth. Any questions on teeth? I'm gonna move on to, I'm kind of done with oral cavity. Swallowing here. So esophagus, I like to talk about swallowing. Let me show you a picture of swallowing, and then let's see if I can cue up a YouTube video here. Hold on a second. Next, right, barium swallow, just so you can see swallowing instead of me talking about something. It's better to see it here. We turn the lights off. I'll try to point out the anatomy you should see. Chew, chew, chew. Look at the soft palate, how flexible it is. Hard palate, soft palate, then you swallow. There's the hyoid, there's your voice box, okay? So yeah, that's swallowing. Now they're drinking. Interesting to look at. If you saw yourself like this, you'd freak out. They're swallowing down the esophagus. That's the next thing I want to talk about. Frontal view. Going down to the gullet, your stomach. So try to follow it up and down from oral cavity down the esophagus to the stomach. You get the idea there. Okay. So the distance between oral cavity and down to here, it's not that long. I'm talking about 10 inches, about the length of the esophagus. I used to have a cadaver where we had the head and only had the esophagus to the stomach. It's not a very large distance from mouth to gullet. Okay, so the esophagus in your neck, anyways, is posterior to the airway. When you swallow, the goal is to push the food to the stomach. Peristalsis is moving it in one direction. But you could have rhythmic contractions where you just mix the food up. But that's not what we're talking about here. When you swallow down the esophagus, it goes down the right tube, it gets to the stomach. So now we've passed through the thoracic cavity into the abdominal cavity. So let's remember where we were, where we're going. Where we're in the oral cavity, you swallow. Here's the skeleton. What we got to remember is the diaphragm is the physical membrane between abdominal cavity and thoracic cavity. Okay, you have three holes here. Boom, boom, boom. that one's for the aorta, that one's for the inferior vena cava, and that one is for the esophagus. To get to the stomach, you gotta go through the membrane there. And your abdominal region has no rib cage around it. So let's kind of build it up there. There's basically your back body wall, your posterior ab wall, with nothing in it. So this picture just adds the artery. You should know this as the abdominal aorta. The abdominal aorta. We're going to study that one in this chapter. It runs from, well, it enters the aortic hiatus down here. That's about the level of what is it? Yeah, T12. Runs from T12 all the way down to around L4 vertebra. That's usually what we say. That's the length of it. Okay. And notice how, well, let's add the artery with the vein in this picture here. Okay, that picture there, we just looked at this picture, all we did was add the vein. That's the inferior vena cava. So you can see the relation. The inferior vena cava is a little bit on the right. The abdominal aorta is a little bit on the left relative to each other. So the full length of the vena cava, the inferior one, is in the abdominal cavity. It has no length in the thoracic cavity. As soon as it enters its hole up in the diaphragm, it enters the heart. The inferior vena cava, we talk about it in terms of how blood flows. Blood is flowing up. The common iliac veins merge to form the inferior vena cava a little lower, right? So they form at about the level of L5. And it runs all the way up higher to T8. So it's longer. So the inferior vena cava is on the right, the abdominal aorta is on the left, and the inferior vena cava is longer simply because its opening in the diaphragm is higher up. Opening up there as opposed to down there. So by that fact alone, it's just longer by default. Now, there's no digestive viscera in here. If you add all the viscera, you get this picture. Covers all this up. This is all the back body wall. Retroperitoneal for the most part. Okay. So when you open an ab cavity, I've done this many times. I just kind of cut along the ab wall, open it up. First thing you see, the fat apron. You can't call it that. What is it? Greater omentum. It attaches to the greater curvature of the stomach. See a bit of stomach, bit of liver. You can see the gallbladder peeking there. It really is that greenish color. Usually on cadavers, that green discolors this entire region. I've even seen cadavers, you can see on top of the skin, it's green because of that. Okay, it's everything green post-mortem. Let's take a break. When we come back, we'll talk about the stomach. Let's take a shorter break. I said you don't have to leave because of the poor air quality today. I'll start up in about 2 minutes. If you want to take a quick bathroom break, go ahead and just go ahead and stay here if you like. I may leave the room momentarily, so don't steal anything.